So our next speaker is Oli Clarke. So Oli is associate professor at Columbia University, and he works on a variety of different biological systems, including Wald particles and ion channels. But he's also pushing the limits of cryo-EM towards the theoretical limits, I guess. So many thanks for coming.
Thanks very much, Anna, and it's really fantastic to be here. It's my first time at CCP-EM, and it's been a really great meeting. Yeah, so what I want to tell you about today is something that's not really related to what we're doing in the lab predominantly. It's more a methods-focused talk, but I felt like maybe this would be a good venue for that. And it really arises from basically an observation we made during data processing. But it's motivated by, I think, an important question, which is, let me just see if I can get a pointer working. Apologies. It's motivated by an important question, which is, sorry. Let me get this working so that things are done. Okay. Is this working? Is this not working? It's fine. Okay, doesn't matter. Basically, small proteins, so proteins less than 50 kilodaltons, are still really challenging targets for cryo-EM. Because for a single particle, difficulty of a structure determination problem decreases with molecular weight. So the decreased signal-to-noise ratio makes particles harder to pick. Denoising helps with that, as we heard earlier. But when we're using the non-denoised particles, the resulting particles are more difficult to align and orient. And so as a result of that, there are only a handful of structures solved by a single particle that are smaller than 50 kilodaltons, and no protein-only structures that are smaller than 30. But there are a lot of really important proteins in this size range, right? If you think about kinases, small enzymes, small membrane proteins, and lots of those are difficult to crystallize. So we don't necessarily have another structure determination technique that will work for them. So if we can come up with new approaches to solve structures of small proteins, it could be kind of handy. The theoretical limit for single particle analysis of proteins in general was estimated by Richard Henderson back in the 90s at 38 kilodaltons. But we already have structures that are solved that are smaller than that. So we know that that is not a hard threshold. We can definitely go below 38 kilodaltons. The question is how far below and how do we do it? And so a key step in and point of failure in structure determination is the estimation of your initial particle orientations during ab initio reconstruction. And so what we were wanting to think about was, maybe this is the point at which things fail. Maybe it's a bottleneck. Maybe we can find improved parameters that work better for small proteins or small macromolecular complexes. And if you think about that in terms of this sort of classic workflow, this classic pipeline for structure determination, maybe it makes a little bit of sense. So normally what we will be starting with is we'll start with a model that these days is typically derived from ab initio reconstruction using stochastic gradient descent. We'll low-pass filter it, usually pretty aggressively, and then we'll compare projections of that model in a certain particle, in a certain orientation with our particles, with our particle stacks. That then allows us to obtain a reconstruction, which will hopefully have improved features, which will let us get a better orientation with better matching projections, get more detail. And the hope is that it's a virtuous cycle in which your orientations improve in accuracy, you get more details in your reconstruction that allow that process to iterate. But if your starting model has insufficient features, your resulting orientation assignments can be ambiguous and end up trapped in false minima. And that is exactly what can happen with smaller particles, because you have, if you think about a 30 kilodalton kinase and you filter it to 30 angstroms, as you might typically do, you really don't have many low resolution features there. It looks like a blob. and different orientations of a blob look pretty similar to one another. But we do know that you've got plenty of high resolution features, right? If you look at 2D classes of something in that size range, if it's in thin ice, if it's well behaved, you will see helices, and in favorable cases, you will see the periodicity of those helices, meaning that you do have high resolution information that's accessible. So we wanted to think about whether we could exploit this. And so Oli has already covered this earlier, but so I won't go through the background, but stochastic gradient descent, which is used for ab initio reconstruction, has typically been applied in resolution increments starting at fairly low resolution and proceeding to medium resolution. So starting at, say, 20 to 30 angstroms and proceeding to, say, 12. We'd noticed, and others had noticed some time ago, that for certain cases like membrane proteins, often it helped to start at somewhat higher resolution, start using information up to somewhat higher resolution, presumably for the reasons that I described. But the way that ab initio reconstruction has typically been used is to generate a map that's used as a 3D reference for subsequent refinement. And all your orientations are chucked out. You discard the orientations, and then you just use that reference, filter it, and use it to correlate with your particles. But as I mentioned, so for small proteins, we often don't have enough features at low resolution to allow for reliable convergence. And so we were curious as to whether incorporating higher resolution information from the get-go and allowing more iterations for it to converge could lead to a better result. But to investigate this, we wanted to have some data sets that we knew didn't work by any other approach. Unfortunately, Gabe Lander and Mark Herzik and Mengyu Wu published this paper, a really beautiful paper some years ago in 2019, where they were looking at exactly the same question in a slightly different size range at the time. So looking at sub 100 kilodalton proteins and solving them by cryo-EM using a 200 kV source. And they solved some beautiful structures, but they also, number one, deposited all their data, which is fantastic. So they put everything on Empire, but they even put on Empire 2 data sets for which they were not able to obtain high resolution reconstructions. One of them was a protein called IPKAC, which is the catalytic domain of protein kinase A bound to a short peptide. And the other is of hemoglobin, where there's a certain portion of the hemoglobin that's split apart into a dimer, so about 28 kilodaltons. And in both of these cases, the data had high resolution features, had beautiful 2D classes with well-defined secondary structure, but they weren't able to obtain high resolution reconstructions in 3D. And in the case of IPKAC, this is around 40 kilodaltons. And when I say it had really good 2D classes, these are the sort of 2D classes that you get. So if you have a close look here, you'll see that the ones highlighted in purple, the good ones, they don't just have helical features. If you look at individual helices, you'll see that those helices have periodicity. They look like corkscrews. So that's already telling you that there is information there that is at least better than 5 angstroms and probably better than 4. And so this suggests that structure determination ought to be possible. I should say also that for getting these kind of, not these ones, for getting these kind of 2D averages, you really need to be performing a lot more iterations during 2D classification than you might typically. And obviously, you can't bin your particles. You need to use particles that are bin to, say 1 angstrom per pixel, not to 4 angstroms per pixel as we might otherwise do. So using this data set initially, we came up after I'd spent quite some time coming back to this data set and trying to get something that would work. I'd tried a lot of different things, nothing had worked. And I came up with this workflow, which we ended up calling HR here. So it's high resolution heterogeneous ab initio reconstruction. And this worked. So I want to go through it step by step. But the key step is ab initio, where basically we use high resolution information from the start. So in this case, we used information up to 5 angstroms to begin with, and then used very, very fine increments up to, in this case, 2.3. And this was in a 3-class ab initio job, just starting from selected 2D classes. And then the difference with a regular workflow also comes in the fact that we don't throw away our particle orientations. So we take both the volume and the particles from this job and just go straight. We reconstruct and then go straight into local refinement using the orientations that we've determined using ab initio reconstruction. So to go through the workflow step by step, so as Ali mentioned earlier, the micrograph denoising really, really helps to see your particles. So training a denoising model on your particles makes a tremendous difference for these small particles in terms of seeing them and accurately identifying their centers, which helps with picking and it helps with everything downstream. For 2D classification, so in this case, we were just using low resolution templates from an initial round of 2D filtered to 20 angstroms. We got 787,000 particles. We put them into 2D. We do 80 iterations of 80 initial iterations of 2D classification, followed by 10 full iterations using a maximum resolution of 3 angstroms. And that gives very nice results. One of the things that I want to emphasize here, that's a lot of iterations. Do you really need that many iterations? So not for all of these classes, some of these classes converge quite quickly, but some of them don't. Some of them only converge really towards the end of classification. So if you have a look at this and you keep your eye on this one in the upper right-hand corner, you'll notice that it really only converges. So it's not converged there. It's not really converged there, boom. So it's only really converged by like iteration 70 something, right? And if that's one of your rare iterations that your rare orientations that you really need to get a reliable ab initio model, you kind of want to separate that out to begin with. Then we perform a 3-class ab initio. In this case, we're using 150,000 particles that we selected by 2D. In this case, we can't really go straight from the picked particles, although I'm going to show another case later that's different to that. And that gives us one good isotropic class, because this is a case where we also have preferred orientation. So we have one very streaky class, one anisotropic class, one junk class, and one nice isotropic class. And what you'll notice is that we already have very clear side chains and actually ligands already at the stage of ab initio reconstruction. And so we can take that, just reconstruct, perform local refinement, and get a 2.7 angstrom map. I should say that's nominal 2.7. At the time, we didn't have a gold standard split available in ab initio, but you can see that the features that are present in the map are, I would say, fairly consistent with a 2.7 angstrom map, right? We see features of our ligand, well-defined phosphates, clear ions, small side chains, and even some carbonyl bumps. And if you have a look at the map overall, you can see that that is pretty pretty consistent overall. Oh, one of the other things that I meant to mention here. So in terms of determining how good a map is, right? Like you can look at overall resolution, that's one thing, but that's a little tricky because, there are issues of overfitting and there are also issues of anisotropy. So just one single number doesn't necessarily tell you what you want to know, right? What you want to know is this map good? Can I interpret it, right? And so to get a measure of that, what we've been doing is we use this wonderful program from Shaw's and Dari, Model Angelo, which is a neural network-based automatic model building approach. And so using that, we can get an idea of, okay, how much of this map can be automatically interpreted? And how much can we build sidechains for? And how much can we build main chain for? And that gives us a fairly good feeling for how interpretable the map is at what resolution. And so in this case, we can automatically build 325 out of 356 residues that are present in the sequence. And the remaining 25 are actually kind of interesting because they're present in an N-terminal helix that is ordered in the crystal structure, but it's totally disordered on map. We see no evidence for that helix whatsoever, right? So it seems to represent something genuine, not just an artifact. But we wanted to have a, oh, right, yeah. So while we were doing this, shortly after, or basically at the same time as we deposited the preprint, Nico Grigoriev's group actually came out with a different approach approaching to exactly the same question using the same data set, but from a very different direction, but also exploiting high resolution signal to get particle orientations rather. And what Nico did was they applied high resolution 2D template matching, which has in the past been applied to very large things with low signal to noise. So for example, ribosomes in fib milled slices of a cell. And they took that and they applied it to IPKAC in order to simultaneously identify where the particles were and what their orientations were. And then they could reconstruct and do local refinement and proceed sort of similarly to what we'd done. And this works really well. The only caveats are that it's very computationally intensive, even more than what we're doing. And it does require that you have a structure to begin with, right? You do need to have a pretty good atomic model. And that's actually great for a lot of cases, right? If you think about structure-based design, you want to solve a bunch of different structures of a kinase, that works great, but it might not be perfect for every situation. The other really neat thing in this paper was that Nico went back and redid an estimation for how small we can go using modern approaches to single particle analysis, and came up with quite a different number of 15 to 17 kilodaltons, which I would say, I think that probably checks out, but we'll see. So we had a look at another example. So we had a look at the hemoglobin dimer. So if you take hemoglobin and you put it on a grid, you mostly get tetramers, right, of the alpha beta heterodimer, dimer of dimers, but you get a certain population of dimers. And those dimers are only 29 kilodaltons. So they're really rather small, and they're smaller and also more heterogeneous than IPKAC. And we were able to use this same approach for this data set using a similar workflow with slightly different parameters. So, denoised, blob picking, 2D, separated out the dimers, repicked the dimers using topaz to get more dimer particles. And then we did 2 rounds of 2-class ab initio using a 5 angstrom to 3 angstrom range. And that gave us a 14,000 particle subset, which we took into homogeneous reconstruction and local refinement to give a map with a nominal resolution of 4 angstroms. So just another example here. So this is starting from 588 movies, so it's not a tremendous amount of data. Patch motion, patch CTF, and then denoising. And I think this is a really dramatic example of just how good the denoising is. It works extremely well. And you can already see here that some particles are bigger than others, and you might be able to actually distinguish the dimers from the tetramers already. And then we do blob picking and 2D. And here you can separate out. So these are your tetramers, these are your dimers. We select the dimers and then go into ab initio, reconstruction, and refinement. We are not able to get quite as many residues built as we were for pKa, which is to be expected. This is lower resolution. It's a tougher target, but we can still automatically build the majority of the structure. And just to show you what the map looks like, so you can see that this is not the best map in the world, but it's at least reasonably isotropic. We have helices, we have bulky side chains, and you will see that we have pretty well-defined heme molecules. So from that perspective, it's for 29 kilodaltons, it's okay. We took a third system, a third data set that's a different type. So this is an RNA protein complex. So this is a 40 kilodalton RNA protein complex, which was originally used for a different methods approach, demonstrating a different methods approach. So this was used as one of the examples in the beautiful paper on blush regularization from Dari and Shores. And so this was thought to be previously intractable. It's 40 kilodaltons and it's got some preferred orientation. And so we took just one tenth of the movies that were available on Empire. So we took 1,200 movies out of the 12,000 from Empire and thought we'd have a go at HR here. We decided to try 2 different approaches in this case. We wanted to see whether we could do this without 2D. And that works actually kind of surprisingly. So we have 740,000 initially picked particles. We put those into 2D. We get 219,000 after 2D that are good. We can do a single class ab initio followed by local refinement, and that gives us a map at 2 angstroms. Or if we take our directly blob-picked particles, put those directly into a single 3-class ab initio job, we get one good class, which, by the way, doesn't look so great. It's got helical features, but not sidechain level features. But if we take that, reconstruct it, and go directly to local refinement, those orientations are good enough to refine. You get a map out of that that is nominally 3 angstroms, and you can see that maybe it's 3, maybe it's 3.5, but it's clearly converged pretty well. given that we had no 2D or anything involved. It's a simple, two-step approach. We wanted to push the limits a little bit, so we tried something that was a little bit too small, which is calmodulin. So we collected some data on calmodulin some time ago. We tried to apply multiple different approaches to this. We haven't got there yet, but what I would note is that in 2D, at least, you can see something that looks like calmodulin, right? This is only 16 kilodaltons, but you can see that we have a clear bilobe structure with maybe, if you've got an imagination, you can almost see a helix connecting the two lobes, maybe. I can't, I don't know. If you're going to ab initio, you can get something that looks like a dumbbell, but we haven't yet been able to take this through to secondary structure kind of resolution. But the fact that we can see subdomain level features in 2D sort of encourages me that in the future it might be possible with better detectors, better algorithms. This was actually collected, I think, on a K2. So this was an old data set that we collected ages ago in a moment of getting a bit out ahead of our skis. But maybe if we collected this with a modern detector in very thin ice, it might be possible. I don't know. Finally, we wanted to sort of go in the other direction and see whether we could apply this to that other low signal to noise regime that I mentioned, which is in situ cryo-EM. So we took a data set from Empire, from Jennifer Lippincott-Schwartz's lab, of just U2OS cells that had been vitrified. And we denoised the micrographs and then picked particles using blob picker and put those through HR hair. And we were able to obtain just from 15,000 particles that were picked from that Empire data set, from a small amount of that Empire data set, we were able to put those directly into HR hair, and we could get a 3.1 angstrom reconstruction of the ribosome. And this had been previously solved by high-resolution 2D template matching, and all the other ab initio parameters, conventional ab initio parameters that I tried on this just did not converge. They just never converged on anything. Interestingly, in this case, it did converge, but it took quite a long time. So just to show you, so it hangs around 400, 800, 1200, 1600, 2000, 2400. Oh, there we go. We've got a ribosome. So it does get there, but it bounces around the landscape for quite some time until it finds the correct minimum. And if you then take that and you perform local refinement, you can get a nice map at 3 angstroms. Okay, so I've gone a little bit fast, I guess, but just to say, so this is currently a preprint available on bioRxiv. We should submit it somewhere. We haven't yet. We're going to. But we've basically made this observation that this seems to be a useful approach for solving small structures. It does have some limitations. So the workflow in CryoSpark at the time that we sort of developed this didn't include a gold standard split. As Oli mentioned earlier, it does now for single class ab initio jobs, and that definitely helps to mitigate overfitting. The run times for some of these jobs can be quite excessive, from a few hours to a day or two. So there's obviously room for further tuning. I would usually choose the resolution limits for ab initio based on what you can see in 2D. But I would say, if it solves your problem, then maybe, go away for a long weekend and you come back and maybe you have a reconstruction. I don't know. And just to point out that, I think Nico's work on high-resolution 2D template matching applied to the same problem is a really nice complementary solution to the same issue. Also, we did try the same workflow in Reliant, and it does work. I couldn't get it to work for all of these data sets, but the same principles can be applied in Reliant. It's just a little more sensitive to the parameters that you choose. And just to acknowledge everybody, so particularly Kukju, who's the first author on the preprint, and Juan and Emmanuel, and everybody else in the Clarke lab, and the NIH and the Irma T. Herschel Trust for funding. And thank you very much for listening, and I'm happy to take questions.